Okay, so as we said in class today, what happens here when you combine an amino acid with another amino acid? You're forming water, but what you're doing is you're forming a dipeptide bond and water. So what you're doing again is you're eliminating water, so it's dehydration synthesis. Or they refer to it as condensation. Okay, so that's going to give you just the first primary structure of proteins. Okay, then we move on to secondary. Okay, secondary structures can form two things. They can form an alpha helix or a beta pleated sheet. So if it's an alpha helix, it kind of looks like a ribbon almost. It's going to be a really bad ribbon, sorry guys. Okay, so what happens here is you kind of have like two dimensions to it. One is one component and then one is a different component. So let's go ahead and show you what this would look like. So this would be one part of the alpha helix, and then it would go down here because it's wrapping itself. It's showing like the inside and then the outside, if you will. Okay, and what happens here is you're going to form hydrogen bonds in between these two pieces. So these are hydrogen bonds. Okay, and if we don't have an alpha helix, so this is an alpha helix, we form what's known as a beta pleated sheet. And that kind of tends to look like this, kind of like a checkered board almost. This is a really bad checkerboard. Sorry guys, it's late. My drawings are not going to be very good right now, and no worries if yours are not either. So I'm just going to draw one for time's sake. Oh no, just kidding. I have to draw two. Okay, this is like not good at all. The top one looks way better than what it should. Now here, you're going to have your hydrogen bonds still form in between these two pieces of amino acids. So this is a beta pleated sheet, so you might see something that looks like this. Sorry, this is not drawn to scale, but hopefully you kind of get what I'm going at. So right here we have your beta pleated sheet, and this whole structure right here would be one amino acid, and then this right here would be one amino acid. So that's what's representative of those small boxes. And again, right here in between, we do have our hydrogen bonds. So always in between those. What's going to happen here is the structure is going to be a polypeptide, meaning several, polypeptide chain, or chains. They form as a result of hydrogen bond. So how they orient themselves, whether it's an alpha helix or a beta pleated sheet, is dependent upon the hydrogen bonding. And that's just the electric attraction between or amongst the amino acids. And we'll get into that, and you guys will see that as we continue on. All right, next is what we refer to as the tertiary structure. So secondary, tertiary. Okay, so tertiary structure It looks a little weird. I'm going to kind of draw it as a globular. It's going to look, you're going to be like, what the heck is she drawing? Just bear with me. Okay, so it tends to take some different types of shapes here. And again, this is dependent upon how it interacts with all the other molecules. So it looks a little funny right now. And then the striations are just to represent that there are molecules there. So it kind of looks like a rope almost. And then it takes kind of almost like a knot position, but it's not fully formed yet. So it's not going to look full or complete to you. Let me find my marker. Okay, so it kind of looks a little bit different. It's just to show you how well these molecules are going to attract to each other to start forming this either globular or fibrous structure. So in the tertiary structure, we have, it's known as the secondary structure. And it's folded more compactly in a compact position. Okay, so what you'll see here is, again, it's dependent upon the electric attraction between them. So if you were to have like a chain, let me draw this a little bit more. These should all be different colors, but for the sake of time, just to represent the amino acids. Dependent upon the structure, so this is going down like maybe in a closer view, you could have a disulfide bond, so a sulfide bound to another sulfide, so that would be a disulfide bond. You could also have, it's not to scale, so please understand that. You could have a carboxyl attached to an amine group, which is going to form a weak bond. That's not a strong bond. Your disulfide bond is going to be strong. And then you can also have a hydroxide bound to, let's see, another oxygen by itself, and that also is a weak bond. So where you're really going to see the strong bonds is in that disulfide bond. So again, this is just showing you it's going to react based upon or combine together to form that globular structure, dependent upon the attraction among the amino acids. And lastly, we have quaternary. Okay, so with quaternary, it's going to form like several proteins. This is so not good. Sorry, guys. So this is 1. This would be 2. This would be 3. And that would be 4. So like this could be an alpha helix 2. This could be a beta 2. This could be an alpha helix 2. This could be a beta 3, dependent upon the structure that it takes. So it is, the quaternary structure is a combination of 2 or more polypeptide chains in a tertiary form, so in a 4. Okay, so it just depended upon what specific protein we're talking about. Now proteins, if we're going to move up from here, and I will take a screenshot of this, so if I'm going too fast or you didn't get all that, I will go into it. I will take a picture of it for you. So proteins can actually become 2 different shapes. they can become globular. Oops. Globular. Sorry, that's globular. And with these, we normally see them as enzymes. And again, enzymes are biological catalysts. Okay, we can also see them in our body, globular or in a globular structure, as antibodies. Okay, and this is our defense mechanism. Okay, we can see them inside our cell membrane as a plasma protein, and those will be used for transport. Okay, we can also see them as hemoglobin. Okay, and what hemoglobin does for you, it's actually 4 compact polypeptide chains with iron containing this heme, which we call iron, heme, at the center. So it's going to have iron in the center, and then it's going to be surrounded by that, the rest of the items. Okay, what else can it do? It actually, in the globular structure, just in of itself, is what we say it's compacted and folded as spherical molecules. Okay, so globular, you just think a glob. All right, other things that we know about globular proteins is that hormones are also found in this category. Okay, hormones are part of the nervous system. And more importantly, we need to know that globular proteins most likely are soluble in water. Okay, so that's all globular. So again, we're thinking of a glob of amino acids. Okay, next, if they don't take on a globular role, they'll take on what we call a fibrous role. Okay, so this is type 1, this is type 2. Okay, and what this kind of looks like is it's like a weaving of the proteins. So they kind of form like multiple ropes among each other. So it's kind of like a long rope, if you will. Okay, so those are like fibrous. Then you can actually show like all the fibers in there if you'd like. All right, so fibrous proteins, they perform for more like structural functions. They contain parallel polypeptides with cross-links, so that's why you can see like that rope-like structure. And these ones are actually insoluble in water. And that is because of them being used structurally. And I'm pretty sure we are good on that. I don't think there's anything else that we need to add. So this is just kind of like a basic overview of what we should know going into building proteins, which will take place on Thursday. And then tomorrow in class we will be going over just proteins in general and making sure we have a clear understanding of what proteins are.